Dear students, welcome to today's botany class on respiration. So today will be our last class on respiration, and coming to the last part of respiration, which is electron transfer system and oxidative phosphorylation or synthesis of ATP molecules. So revising the components of electron transfer system. So here we see this side is the mitochondrial matrix side, this is the intermembrane space, and this layer represents our inner membrane. So this is the outer membrane part of the mitochondria. So this part is enlarged in the figure, showing you the mitochondrial matrix part, the inner membrane, the intermembrane space, and the outer membrane. So as we know, the membranes are phospholipid bilayers which are present, and these are the electron transport carriers or the ETC chain present on the inner membrane. So revising the components of the electron transport system, the first complex, so this is complex number 1. So this complex is NADH coenzyme Q or ubiquinone oxidoreductase, also represented as NADH dehydrogenase. Then this is the second complex. This complex is known as succinate dehydrogenase. So this complex succinate dehydrogenase is present also in the Krebs cycle, where succinate is converted into fumarate. So the succinate dehydrogenase enzyme is present towards the matrix side of the inner membrane. So this you have to remember. So this is succinate dehydrogenase is the second complex. Then the third complex is cytochrome BC1 complex or cytochrome C oxidoreductase. So this is the third complex which is present in the inner membrane. Then comes the fourth complex or complex 4, which is cytochrome C oxidase. So this is the last fourth complex that is present. So in addition to these 4 complexes, we find 2 mobile carriers. One is coenzyme Q or ubiquinone component, and the other is cytochrome C. So these 2 components, which is ubiquinone and coenzyme Q and the cytochrome C, they are called as mobile carriers. So coming to the characteristic feature of these complexes, the complex of NADH coenzyme Q oxidoreductase has flavin mononucleotide as a prosthetic group and also iron sulfur centers. So the second is succinate dehydrogenase, which also has a type of flavoprotein which is FAD or flavin adenine dinucleotide. It has an adenine component. And the third one is complex 3, which is cytochrome BC1 complex. So cytochrome complex has a number of cytochromes, namely cytochrome B and C1, and here also there is the heme component or iron component that gets oxidized and reduced. In the fourth complex, we have complex 4 as cytochrome C oxidase, where A and A3 are the components and copper and iron centers are present as the oxidation and reduction centers. So we can see how the electron transport occurs in these 4 complexes in the next slide. So electron flow in the ETS, if you go to the type of electron flow that will occur in the ETS, suppose this is the inner membrane. If this is the inner membrane, we can also represent the complexes. The first complex which we took is, this is the first complex that was present. Then the second complex on the inner membrane towards the matrix side. Then we have the third complex. Then we have the fourth, and then the ATPase component in the membrane. So, coming to the characteristic feature of the first complex. So, this side is the intermembrane space. Intermembrane space. This side is our matrix. This is inner membrane, and here you have the outer membrane. So, what happens in these structures is the prosthetic group here that is present is FMN or flavin mononucleotide. This is FMN. The complex is NADH dehydrogenase or NADH coenzyme Q oxidoreductase. So, here NADH will be converted into NAD plus in the matrix. So, NADH, which was formed during the Krebs cycle or the citric acid cycle and is present in the matrix, it donates its electron. So, NADH has a redox potential of minus 0.32, and so it easily donates an electron to an electron acceptor. So, the electron acceptor in the first complex here is flavin mononucleotide, and then this electron, there is movement of 2 electrons from NADH in the form of the hydride ion. So, the hydride ion is present that takes up the electron and then it gives it to FMN. So, FMN is now reduced. So, what happens when it gains electron? FMN is reduced. Reduced FMN donates the electron to a iron sulfur center. So, in the iron sulfur center, the Fe3 plus component gets changed to Fe2 plus by gaining electrons. So, this is the iron sulfur center present in complex number 1. So, this complex number 1, NADH coenzyme Q oxidoreductase or NADH dehydrogenase gains electron from NADH that was formed from the Krebs cycle. So, from here the electron from the iron sulfur center is donated to the mobile carrier ubiquinone or coenzyme Q. So, ubiquinone on receiving electron is reduced by taking up 2 H plus ions from the matrix and it gets converted to UQH2. So, this is the reduced form of ubiquinone, also known as ubiquinol. So, from here, so this is now the mobile carrier. So, this is a mobile carrier ubiquinone that takes up electron. So, it gets reduced. So, while this movement is taking place, then the electron free energy also helps in the movement of 4 H plus ions towards the intermembrane space. So, 4 H plus ions are moving towards the intermembrane space from complex 1. So, this complex 1 is a proton pump. So, it is pumping out H plus ions from the matrix side towards the side of the intermembrane space. So, when electrons move to ubiquinone, then ubiquinone takes up H plus from the matrix side, gets reduced to UQH2, and then it transfers the electron to the third complex. So, this is the third complex. So, here we are seeing the movement of electrons first from NADH, then we shall see the second complex movement from FADH. So, here this is the third complex. The third complex is having cytochrome B and cytochrome C1 with iron sulfur centers. So, heme part here is the receiving end of electron and it contains iron and sulfur component. So, here the electron moves to cytochrome B. From cytochrome B, the electron is transported to cytochrome C1 through the iron sulfur center. So, during this movement of electrons also, 4H plus are moving towards the matrix side. 4H plus move towards the intermembrane space, 2H plus from the matrix side and 2H plus from the reduced ubiquinone or ubiquinol that is present in the layer. So, this ubiquinone is mobile, that is it is able to move inside the inner layer and it is a lipid soluble molecule. So, here 4H plus ions are again moving towards the intermembrane space. Then from the cytochrome C1, the electron moves to another mobile carrier. This mobile carrier is cytochrome C. So, it is not a complex, but the mobile carrier cytochrome C. So, cytochrome C is present in the inner membrane towards the outer side or towards the intermembrane space. From cytochrome C, the electrons pass on to the fourth complex. So, here electrons are moving from higher redox potential towards a lower redox potential in accordingly how the complexes are arranged in the inner membrane. So, here from cytochrome C, the electron is taken up by cytochrome A, and then from cytochrome A, the electrons are transported to cytochrome A3. So, in this complex which is cytochrome C oxidase, so here there are two reducing and oxidizing centers, one is iron and also the copper centers. So, these two get reduced and oxidized in the complex 4. So, here again Fe3 plus gaining electron becomes Fe2 plus, then Cu2 plus gaining electron becomes Cu plus form. So, cytochrome A and cytochrome A3 are the only two components that can transfer these electrons to oxygen. So, where the electrons will go? The electrons will move into the matrix, they will be accepted by the oxygen. So, here oxygen molecule is going to receive the electrons moving in the transport system. So, here we can write half O2 plus 2E minus or 2 electrons gaining the protons from the matrix, you get a molecule of water. So, here this is how water is formed and this is terminal oxidation. So, here cytochrome A, cytochrome A3 and the acceptor, terminal acceptor for the electron is the oxygen molecule. So, this is formation of water. During this movement of electron in the inner transport system, here again 2 H plus ions are transported into the intermembrane space. So, here we are finding that in the electron flow in the ETS, there are two things that are happening. One is the movement of protons from or H plus ions from the matrix side towards the intermembrane space. So, this is when the NADH donates its electron to the first complex and then through the mobile carrier to the third and then to the fourth. The two mobile carriers are ubiquinone and cytochrome C. Then what happens to complex 2? This complex 2 present towards the matrix side of the inner membrane is succinate dehydrogenase. Is succinate dehydrogenase. So, this enzyme was also taking part directly in the Krebs cycle, the conversion of succinate into fumarate. So, in this step we saw the synthesis of one molecule of FADH2. So, here the prosthetic group will be FAD. So, FAD molecule will get converted into FADH2 or the electron will go from here to FAD and then from FAD again to ubiquinone or coenzyme Q. But you have to remember that FAD molecule here is this complex is not an proton pump. The proton pumps are three complexes, the complex 1, the complex 3 and the complex 4. This complex of succinate dehydrogenase does not pump any proton to the intermembrane space. So, this is how electrons pass through the electron flow in the ETS or electron transport system. So, this structure here at the end of the electron transport is your F0 F1 particle or ATP synthase, also sometimes known as the fifth complex in the membrane. So, it helps in the synthesis of your ATP molecule. So, first we shall go to the balance of H plus ions on both the sides by the chemiosmotic hypothesis. We shall see the structure of the ATP synthase and then how it is again formed. So, after the electron has been transported and then the H plus ions also transported into the intermembrane space, we go for oxidative phosphorylation or the synthesis of ATP molecule, because our primary aim in respiration is the synthesis of ATP molecule. So, in the electron transport system, oxygen was the terminal acceptor of the electron, and during the flow of these electrons in the electron transport system, we saw steps also H plus ions or protons were transported into the intermembrane space. Then how is ATP synthesized? So, the method of synthesis of ATP is also called oxidative phosphorylation, because the oxidation of the compounds results in the synthesis of ATP molecule, whereas comparing the synthesis of ATP in the chloroplast, that was photophosphorylation, or light was responsible for synthesis of ATP. So, this process here again is explained by the chemiosmotic hypothesis of ATP synthesis, and it was proposed by British biochemist Peter Mitchell in 1961. He was also later awarded the Nobel Prize for finding out the chemiosmotic synthesis of ATP molecule. So, the electron transfer in the inner mitochondrial membrane, as we saw, results in proton translocation from the matrix So, we saw the movement from the matrix towards the intermembrane space in the last slide, or in the electron transport system. Then what happens after this is, we find that these protons will be moving towards the intermembrane space. So, the pH of the matrix will then increase to 8, that is, we have loss of H plus ions. So, the pH increases up to 8. So, the acidic condition is not present, and we get it in the alkaline form, that is in the pH of 8. So, in the intermembrane space, however, the pH reduces to 7, and we have a pH difference of about 1 unit between these two structures. So, here you can see in the figure, so this is the mitochondrial side. So, here you see the inner mitochondrial membrane, this is the matrix, and these are the F0, F1 particles present in the membrane. So, the movement of H plus ions from the matrix towards the intermembrane space results in the pH of 8 in the matrix and pH of 7 in the intermembrane space. So, what there is now a difference. So, there is a difference of pH. Then more positive ions, that is H plus ions, are positively charged. So, more positive ions are now present in the exterior phase of the inner membrane. So, this is the inner membrane, this is the exterior phase or the intermembrane space. So, there we have more of positive ions, and we find negative ions in the matrix phase. So, this difference is also a difference in the electrical charge across the membrane, and this is known as the membrane potential or the electrical potential. So, first there is a difference in the pH, then there is also now a difference in the electrical potential or membrane potential. So, difference in both the pH as well as electric potential, they contribute to the development of what is called as the proton motive force. So, this is development of proton motive force. So, this proton motive force will be responsible for pushing back the H plus ions from the intermembrane space back into the matrix. So, through the F0, F1 complex, and this movement will help in the synthesis of ATP. So, we shall see how there is synthesis of ATP by first knowing the structure of the ATPase or F0, F1 component. So, here you can see this component, which is the F0, F1 component, and here this is the F component, which is called the stalk, and this is the head component, which is called as the F1 component, having enzyme ATP synthase. So, this side we have more of H plus, and this side there is less of H plus that is present. So, here we can see the structure of the ATP synthase or F0, F1 component. So, ATP synthase or F0, F1 component uses the proton gradient energy for the synthesis of ATP. So, this proton gradient is the difference in the H plus is a proton. So, the difference in the intermembrane space and the matrix will help in the synthesis of ATP. So, what we saw in the last diagram was this ATP synthase is a large transmembrane protein complex. A transmembrane protein complex is a protein complex that will be traversing the entire inner membrane. So, if this is the inner membrane, we see this is moving from one side of the membrane to the other side of the membrane, projecting into the matrix. So, it is a transmembrane protein. So, this structure is composed of a knob and a stalk-like structure. The F0 component is the stalk that is embedded in the membrane, whereas the F1 component is the knob or the head. It is the knob or the head that is present. So, here this is the F0 component, this is the F1 component. So, here this F1 or F0 stalk has a proton channel. It has a proton channel for the movement of protons across the stalk. So, it spans the membrane. So, here you can see it is inside the membrane. Then it forms a proton pore, and the membrane spanning portion is integral membrane protein. So, it is integrated inside the membrane, and it is made up of 4 different types of subunits. So, it is made of 4 different types of subunit, and next comes the F1 knob. So, F1 knob, this is the F1 knob component of ATP synthase and contains the catalytic subunits or ATP synthesizing subunits. So, synthesis of ATP is mainly by the help of the knob or the F1 component. So, here what happens is ATP synthesis takes place. F1 knobs faces the inner phase of the inner mitochondrial membrane towards the matrix. So, here you have different components alpha, beta, delta and gamma components that are present. So, here also you can see alpha, beta, gamma and delta components present. So, passage of protons through the F0 or stalk into the matrix is coupled to ATP formation, and it is estimated that the passage of 3 H plus ions through this knob will be synthesizing one molecule of ATP. So, how ATP is synthesized? So, F1 F0 complex will serve as the molecular apparatus for coupling H plus movement to ATP synthase. So, passage of proton through the F0 channel will cause the rotor to spin in one direction and the starter to spin in the opposite direction. So, you can imagine this component or the F1 component to be like a motor or a hydroelectric power station. So, when this rotates, there it brings into the activation of ATP synthase. So, the unit rotates, there is conformational change in the head part or the F1 component, and then ATP synthase helps in the synthesis of the ATP molecule by combining ADP and inorganic phosphate present in the matrix. So, for the movement of 3 H plus ions, there will be approximately synthesis of one molecule of ATP for each 3 plus ions moving across the structure. So, in the electron transport system, we saw that this F0 F1 component is present towards the end after electron transport is occurring. So, after electron transport is occurring, then we find that there is movement, there is chemiosmotic movement. So, here these protons will be moving. So, when these protons move, there is movement of H plus ions first through the intermembrane space, then it goes to the F0, through the F0 it passes on to the F1 component of the stalk. So, here you can again see that here from NADH 4H plus were moving or passing to the intermembrane space, and from the second complex, which is not a proton pump, there is no movement of protons towards the intermembrane space. In the third complex, which was cytochrome BC1 complex, there again 4H plus were transported to the intermembrane space, and in the fourth type complex also there was movement of 2H plus ions into the intermembrane space. So, here from NADH the number of H plus ions that we are going to get is 10 H plus ions. So, 10 H plus ions are approximately released when NADH dehydrogenase takes up electron. So, these protons will be then directed towards the F0 F1 component. So, there will be movement in this direction. There will be rotation or conformational change of the F1 component, and these H plus will be directed into the matrix. These H plus ions will never be directed directly from across the intermembrane space into the matrix side. So, here when there is movement of 3 H plus ions, there will be synthesis of one molecule of ATP by combining ADP and inorganic phosphate. Similarly, in case of FADH2 or FAD or the second complex, we can find the number of protons that are released is not 10, but it is reduced because from here the FADH2 is getting the electrons. So, it is only 6 protons that are released. So, approximate calculation is 10 protons moving through the ATP synthase will be giving you 3 ATP molecules, and 6 H plus ions will be giving you 2 ATP molecules. So, calculating how much of ATP can be synthesized by aerobic respiration of glucose. So, coming to first the Krebs cycle. So, in Krebs cycle, the various steps released different number of NADH. One, from the conversion of pyruvate into acetyl coenzyme A. So, conversion of pyruvate to acetyl coenzyme A, that is synthesis of 1 NADH plus H plus. Then again, during the synthesis of alpha ketogluteric acid, we have another NADH molecule that is produced. So, all oxidative decarboxylation reactions were releasing NADH molecules. Then to succinate. So, there again NADH molecule was released, and the fourth NADH was released during the conversion of malate into oxaloacetate. So, the number of NADH molecules released from Krebs cycle is 4, and glucose breaks into 2 molecules of pyruvate. So, for 4 molecules of NADH, the number of ATPs that can be synthesized will be 4 into 3 equal to 12 ATP. Similarly, for FADH2, the number of ATP, the FADH2 was produced was 1. So, FADH2 will be giving you 1 into 2 ATP molecules, that is 2 ATP molecule during the conversion of succinate into fumarate. So, you will be getting 1 molecule of FADH2 getting 2 ATP molecule. There was also synthesis of 1 molecule of direct GTP, which is converted into ATP when the molecule of succinyl coenzyme A broke into and formed the succinate. So, here again from Krebs cycle you get 1 direct ATP which is equal to 1 ATP molecule. So, from Krebs cycle, the number of ATP that you can derive for 1 pyruvate molecule is approximately 15 ATP molecules. So, from glucose we are getting 2 pyruvate. So, the number of ATPs that can be got is 15 into 2 pyruvate, that is equal to 30 ATP molecules. So, from where does the extra ATPs come? That is from the NADH molecule that were synthesized during glycolysis process. So, in the glycolysis process also we saw there was synthesis of 2 molecules of NADH, and these 2 molecules of NADH are transported into the mitochondrial matrix by 2 different shuttles or 2 different compounds. One is glyceraldehyde 3-phosphate shuttle, which releases the NADH hydrogen from complex number 2. So, it releases it to FAD, so it becomes FADH2. So, when it is moving by the glycerol 3-phosphate shuttle, the number of ATP that can be got from one NADH will be again 2 ATP molecule. But when the NADH is transported by the malate aspartate shuttle, bringing it to the matrix, then again it moves from complex number 1, and so you get additional 6 ATP molecule. So, if we take into account the ATP from glycolysis NADH, so here you can get, if it is 2 NADH entering from first complex, it will be into 3 ATP. So, you get 6 ATP molecules. So, this becomes 36 ATP molecule, and from glycolysis we also had a net gain of 2 direct ATP molecules. So, this is again 2 ATP molecule, and total can be 38 maximum ATPs from one molecule of glucose. So, which can be seen in the heart and liver cells, but generally we get 36 ATP molecule because the NADH from glycolysis is transported by glycerol 3-phosphate, where 2 ATPs are synthesized from one NADH molecule. So, this is how ATP is synthesized during the aerobic respiration process, where oxygen is the terminal acceptor of electron, and the ATP synthase or F0, F1 component in the electron transport system helps in the synthesis of the ATP molecules. So, this was the end of how aerobic respiration was occurring. So, these phases, that is electron transport system and the Krebs cycle, do not operate in the aerobic respiration, and so the energy liberated is only 2 ATP molecule that was synthesized from the glycolysis phase in the cytoplasm. So, coming to the next topic, which is what is respiratory quotient and respiratory substrates. So, respiratory substrates in respiration are the substances that are broken to release energy, and respiratory quotient is abbreviated as RQ and is also known as the respiratory ratio. So, what is the respiratory ratio? It is defined as the ratio of volume of carbon dioxide evolved to the volume of oxygen consumed in respiration per unit time per weight at a standard temperature and pressure. So, simply saying RQ is the volume of carbon dioxide that is evolved to the volume of oxygen that is consumed in respiration per unit time per unit weight. So, respiratory substrates, in respiratory substrates we see the carbohydrates are the most common respiratory substrates, and then they are followed by lipids and proteins. So, the RQ formula will be the volume of carbon dioxide divided by the volume of oxygen consumed. So, how much of oxygen you are taking in and how much of volume of carbon dioxide is evolved is the respiratory quotient. So, respiratory quotient for different respiratory substrates can be RQ for carbohydrates. If it is aerobic respiration, it can be C6H12O6, 6O2, 6CO2 is evolved. So, the RQ will be 6 carbon dioxide evolved to 6 oxygen consumed. So, here the RQ is 1 for a carbohydrate molecule like glucose. So, for the other components, for example, for the fats or lipids, the RQ has been determined to be approximately 0.7. So, the RQ for organic acids like malate is around 1.3. So, this is the respiratory quotient. So, respiratory quotient shows you how much of carbon dioxide is evolved for how much of oxygen is consumed. So, this brings the end of the respiration chapter, and so you can go through the different processes that occur in the respiration, and mainly the Krebs cycle, glycolysis and the electron transport system. So, in the next class we shall start a new chapter on plant growth regulators. So, how plant growth is regulated by different phytohormones or the plant growth regulators.